Anna Rakowski, and I am the co-founder of Cure CMD. And we have the pleasure today of having Dr. Gisele Bonne from the Institute of Myology going over with us her recent work on gene therapy approaches for the laminopathies. And without further ado, Dr. Bonne.
Many thanks, Anne, for the nice introduction. So let's start. So to better understand this webinar, it is important that you know the definitions of some basic scientific vocabulary. So the nucleus is a part of your cells when your DNA is stored. DNA is a language that all our genetic code is written in. A gene is a short piece of DNA, and gene makes protein. In order to go from DNA to proteins, the DNA must be first converted into RNA or transcript, and then produce a protein. Cardiac is a medical word for heart. Skeletal muscle is a medical word for muscles of your body that you can move, biceps, triceps, quadriceps, for example. And murine is a scientific word for mouse. Most research requires a mouse model to better model the human disease. So with that said, let's start. This may look like an architectural diagram. However, it is the way scientists and doctors talk about and present genes and proteins. The LMNA gene has 12 coding regions called exons that are depicted here as boxes. Exons are separated from each other by introns that are depicted as lines on this scheme. Each of our cells in our body contain a nucleus, as I said. Inside the nucleus is the DNA, which is the body's genetic information. While we are eating, walking, sleeping, an incredible process is constantly taking place that helps our cells make the proteins the cells need to stay alive. And to go from the genetic language, the DNA, to a protein, the genetic language must be first translated in RNA, a different genetic language. RNA is then, as I said, transcribed into protein. In summary, this slide tells us that LMNA is a gene. The DNA that encodes this gene is found in the nucleus in all our cells. This DNA is first converted in RNA and then in two different proteins called lamine A and lamine C that are present here in this scheme. And mutation in LMNA gene produce abnormal lamine A and lamine C protein, and in turn lead to the development of the disease, the laminopathies. So, what do lamine A and lamine C in the normal cells and tissues? Before to answer this question, we need to better define what is the nucleus and its envelope. On this slide, we put a scheme of a cell you may probably remember from your high school biology classes. Every cell in our body has a cell membrane and contains a nucleus that also have a membrane and separate the nucleus content from the rest of the cells, which is called also the cytoplasm. If we enter a bit deeper into the cells, on this scheme you have a nucleus with its envelope here that is made of two membranes, as you can see here, the outer membrane and the inner membrane, with inside the DNA. This nuclear envelope has also some holes, the nuclear pore here, that allow molecules to go in and out the nucleus. And this is common to all the cells that compose our body. Now, if we focus on the cells that compose our muscles and we zoom into the structure of our muscles. The muscle cells have a particular structure as they form longer fibers that align in parallel and form the whole muscle. On this image, taken under a microscope, you see a muscle cell with inside its nucleus. And if we zoom in again, its envelope can be present as it is here on this scheme, with its two membranes and the pore. And this nuclear envelope is composed of numerous proteins that are specifically located here and has specific functions. These proteins are either anchored in the membrane, like those ones, like MAN1, MRIN, etc., or they form also a net here that lies just underneath the inner nuclear membrane. This net, this meshwork, is formed of lamin A and lamin C, the two proteins encoded by the LMNA gene. And as we are now inside the nucleus, you have here the DNA. So in summary, lamin A and lamin C, the two proteins produced by the LMNA genes, are proteins of the nuclear envelope. The next question is, how does a mutated nuclear envelope, a mutated nuclear envelope protein lead to a muscle and heart and breathing problem, as it is what happened in laminopathies? What we have learned from our research is that when we look at the skin cells under a microscope and we use specific tools to stain the DNA, here in blue, or the protein lamin A and lamin C in red here, this is what we can see. In cells from a control individual, the nucleus has a nice ovoid shape, as you can see here. In cells from a patient that carry a mutation in LMNA gene, you can easily see that the nucleus is no more normal shape, has numerous bleps, or the lamin organization staining is not as homogeneous as what you see in control cells. So, what we observe is that LMNA mutation lead to abnormal weak nuclear envelope. And this, when it happens in the whole muscle cells of our body, there's muscles including the heart that are constantly under contraction, as the heart beats constantly and you contract your muscle to be able to stand and to move. So when the nuclear envelope of all your muscle cells are abnormally weak, it turns out that the whole muscles of your body became also abnormally weak. And this is roughly how we can explain in simple words how the lamin AC gene defect produces the disease, although the whole mechanism at play are far more complicated. Now that we have explained what are LMNA, lamin AC, and how abnormal lamin A and C lead to the laminopathy disease, we need to define what is gene therapy. Gene therapy aims at bringing to each cell the piece of DNA that will rescue the DNA or gene defect that is present in each cell. To do so, a piece of DNA are first to enter the cells, that is to pass the cell membrane here, to enter the nucleus enrich the DNA. And to do that, we use a vehicle to transfer the DNA up to inside the nucleus. And this vehicle, this vector, is a virus, as viruses has all the necessary machinery to do this job. Of course, we use modified viruses that are not dangerous, the so-called adeno-associated viruses, or AAV. And in the case of laminopathy, what we do want to do is ideally to bring the corrected version of the LMNA gene packaged in viruses to enter each cell of the muscles. So, why a corrected version of LMNA gene? Should an injection of a normal LMNA gene be enough? Well, let's explain the slight difficulty of the laminopathy context. So, I explain you the gene produce the protein that have certain function and structure in the muscle. Okay? In a person unaffected by LMNA mutation, the gene is present in two normal copies that produce completely normal lamin A and C. And we observe normal muscle structure and function as on this picture of a muscle of a normal mouse. Well, in a person affected by a LMNA-related muscular dystrophy, one copy of LMNA is normal, but the second copy is mutated. The protein produced by the mutated copy are mutant and can be partially degraded, and this degradation can be variable. Therefore, this leads to an overall decreased quantity of lamin with, in addition, the presence of mutant lamin in the muscle that can have toxic effect. And thus, the resulting muscle looks abnormal, as here on this picture of a muscle from mutated mouse, with small muscle cells compared to the normal above. And more importantly, this abnormal muscle is not able to correctly function. So we need to remove the mutant protein that exert abnormal function and compensate at the same time the decrease of normal protein. To sum up, gene therapy for LMNA will be only successful if, on one hand, we can administer a normal gene. This may be through a viral vector called AAV, and we are able to fix the mutated lamin A and C protein being made, and for this we will use transplicing, and I will come back on the definition in a moment. Another way to explain this would be that if LMNA affected a computer, you will need to first install a new hardware system on your computer and write a program to fix the old hardware glitch and prevent it from causing problems while a new hardware system launches. And as I just said, we use Transplacer Approach to fix the old hardware glitches. Indeed, you remember, in the normal situation, the LMNA gene is transcribed into RNA or transcript first, then produce the lamin A and lamin C protein. Here I just present the lamin A for simplicity. When you have a mutation, for example here in exon 1, you have the mutation everywhere, okay? And here in the protein, the mutant protein. What we tend to do with transplicing is a kind of surgery of the RNA. We have here, for example, still this exon 1 mutation, and in order to correct it, we will modify the step of producing the RNA, as this step is made of attaching to each other each exon and removing intron that is a line, as you can see here. So during this step, what we are doing, we bring a so-called transplicing molecule that will bring this orange exon and use the remaining of the gene that are not mutated. And this will end up with this mixed RNA transcript that is corrected and do not carry anymore the mutation. And this transcript will be able thus to produce the corrected protein. So also this said this way, it might look quite simple. In real life, it is rather complex to identify the correct working transplicing molecules. So, what we've done since 2 years? We developed, tested, and validated the transplicing molecules. We have produced the corresponding LMNA AAV gene therapy vectors. We have started the evaluation of the transplicing event in normal culture muscle cells, and we have evaluated the potential toxicity of these transplicing events in normal mouse model. What we still need to achieve, and this is our plan for next year, is to improve the technical detection of transplicing events first, and then evaluate the transplicing in mice with LMNA congenital muscular dystrophy. That is, this mouse model that we have developed and generate that carry one mutation in the laminate gene and that show a huge defect and died prematurely, and show this abnormal muscle structure and function. So we aim at looking at if we improve the survival of those mice and their muscle function and structure. In summary, we have spent 2 years working on developing transplicing techniques and gene therapy vectors. In the next year, we will test transplicing in the LCMD mouse model. And this work will tell us if transplicing and gene therapy improves survival and muscle strength and whether it is toxic in the mice. Finally, I will thank the patients and their family, first of all. Also, thanks Feriel and Anne Bertrand. You just seen on the previous slide. Feriel is doing the whole work on this project. And finally, I will thank our funding support, Cure CMD, the AFM Téléthon, and the Andrés Marcio Foundations, as well as our institutional support, the University Pierre et Marie Curie, INSERM, CNRS, and the Myology Institute. And I thank you for your attention.
Great. Thank you so much, Dr. Bonne, for this fantastic presentation. I would like to just recap some of the key points because you've covered quite a bit of information. So what I've heard as some of the key points are that LMNA is a gene. The LMNA gene makes an LMNA protein. And when there is a change in the LMNA gene, the LMNA protein doesn't work very well. And when the LMNA protein doesn't work well, you have LMNA muscle disease, which leads to breathing, heart problems, and weakness. and that you have been working in the last 2 years on 2 different approaches to gene therapy. The first is AAV, and the second is transplicing. That you hope to have answers to whether these 2 approaches improve survival in the mouse model in the next year. I wanted to ask you just 1 or 2 questions. The first question is if you could explain to the audience what the role of using mouse models is, and the second, if you could maybe put your research in the larger context of how long it takes to get to clinical trials if indeed this work shows promise and shows an improvement in survival in the mouse model.
Well, so first, the needs and why we use mouse model. This is your first question. So, yes. Why we do use mouse model is that we cannot, although we have access to patient cells that are currently either skin cells or muscle cells taken along during biopsies, we can explore them as I showed you on the nuclear shape, etc. But those materials, although very precious and very important for us, cannot allow us to explore the function of the muscles and how we can understand how they are not working correctly on one side, but once we try to treat them, we cannot check whether we improve their function. A cell in a petri dish cannot answer those questions. So this is the reason why we have created this mouse model that reproduce mutation identified in patients. And the mouse model mimic, recapitulate in a way, the feature that we observe in patients. And this is like this, that we can understand how having abnormal shaped nuclei in our muscle cells could alter the muscle function, and once we try to correct them, we can monitor how efficient we are regarding the restoration of the muscle function. So the mouse model is essential in our research progress because, as I just said, even if we have access to a few patient cell lines, which are essential for our understanding, this is far not sufficient to go further in the validation of therapeutical approaches. And everybody would understand that we will not test directly our therapeutic approaches directly in patients. We need to validate it in a whole organism, whether it is efficient or not, before to go for patients. And the second question was, how long does it take to go forward a clinical trial if we are lucky enough to validate the validity of our approach we are currently developing? Well, this is also a very long process. What we have done so far is that we have been able to see whether it's working well in a petri dish, in cell culture. Now we start to move towards the mouse model to see whether we are able to modulate and restore the muscle function and structure. But obviously a mouse is not a human. So regarding regulatory authority, before to go for clinical trial, we will have to go over numerous steps from a regulatory point of view. First, to show them, as it is done for any other gene therapy, that the vehicle we use, the viruses, the AAV vector we use, are not dangerous for the patient and do not produce adverse effects. So this is another part of the research that we need to develop. And then, once this will have been done, we need to fill out a lot of files with regulatory authority and go for clinical trial. But from the proof of concept in a petri dish or even in a mouse model towards a clinical trial in patient, I need to say that it will take several years, as it's currently going on for other muscular dystrophy like Duchenne, for example.
Thank you. That is fantastic, Gisele, and thank you so much for your presentation today, and we look forward to getting an update in a year.
Thanks a lot, Anne, for your time and for giving me the opportunity to present our current research and project. And I hope that I will be able in a year from now to show you very good data based on our preliminary results. Thanks again for the support and for the help.
Great. Thank you so much. Okay. Bye-bye.